Well, hello, we're team number 4, and today we're going to explain the comprehension and analysis activity. I'm Berenice, and these are my teammates, Emilio and Danna, and we're going to start with the introduction. So basically what we have to do in this activity is the transcription and translation, of a DNA chain to an RNA, so we can decode secret messages. And now we're going to go to the develop of vocabulary. We have 3 different concepts to analyze, and it's the synthesis, the transcription, and the translation. So first of all, we have the synthesis, which is pretty basic, and it's the process of combining 2 or more objects in a new one. The transcription, it's more related to the use of DNA segments as a pattern, so we can build a complementary chain of RNA. So they are like bonds in a transcription process. It is used to describe one of 2 parts which complement each other, and it contains the necessary information to build the other part, which is the last part of the decode message. And the translation, it's the decoding store instructions in RNA to build a protein use amino acids correspond. And it's clearly defined the relation between a codon of RNA and amino acids. Now we're going to explain briefly the process to decode the messages. And first we have a DNA chain, and we have a code message there, which is built by codons. These codons are basically built by the nucleotides of DNA, which we already know. They're adenine, thymine, guanine, and cytosine. And we are going to decode them into, well, first we're going to pass them to a format so we could fit the RNA standards, which are another nucleotides. We already know them, which are the adenine, guanine, cytosine, but instead of the thymine, it's the uracil. So we decode that DNA chain into an RNA format by the messenger RNA. And then we have to do like the same, but in the inverse way. And that part is related to the transfer RNA. So when we have that one, the last step is just basically to with the codons, we are going to build proteins based on the amino acids. And that last step is when we check another part of information, so we can get the names of the proteins we're going to use and how are they useful for all the process. And lastly, we have what is the role that codons play in protein synthesis, what is used in coded messages. So each codon basically specifies one amino acid. I already told you that they have a relationship because basically a codon, which is built by the nucleotides, is an amino acid. But most amino acids are encoded by multiple codons. So it's a whole, like amino acid, it's the external wall constituted by various codons. So in a reading frame, in an uninterrupted sequence of codons in RNA form a specific from a specific start codon to a stop codon is translated into the linear sequence of amino acids in a protein. So we have the message, and we basically follow. If we follow the steps correctly, finally we're gonna get from the amino acids a protein. And codons provide the key that allows these 2 languages to be translated to each other. And each codon corresponds to a single amino acid or a stop signal, which is it's accepted too. And the full set of codons is what that becomes a protein finally. Now we're going to pass to the decoding of messages, and this is going to be explained by my partner, Danna.
Okay, so to understand everything that my partner Bede has already explained, we're going to start with this decodifying this message. So basically here we are translating. This is the whole process of the synthesis of proteins, as we already explained. So that here we have like 3 main points that we have to remember and keep update. We have messenger RNA, we have transfer RNA or tRNA, and then we have amino acids, which is what the double A stands for. First, we have the code that the first code that we have, like the one that we're going to change and translate, is the one is in DNA format. So the thing with this code is that we can work with the DNA format because translation works with RNA. So what we're going to be doing is that the messenger RNA, well, basically, we're just passing this message to the to the RNA language. And as I wrote in here, we have adenine, which is paired with timing, but in this case, as my partner mentioned, we're not going to be using timing, we're going to be using uracil instead. So the first thing we do is that we take the DNA code. And as you can see, first we have the first column that we have is T, G, and A, which stands T stands for timing, G stands for guanine, and A stands for adenine. So remember that timing is paired with adenine. So that's why in the first point that we have mRNA, we change the T for an A. And then we have guanine on the original message. And guanine is paired with cytosine. So that's why we wrote the C. And then adenine is paired with thymine. But remember that we're not using thymine, we're using uracil. So that's why we wrote the U. And for example, the second one will be CGA on the original message. And we're going to decode and translate it. And so we have cytosine, cytosine is paired with guanine. So that's why we wrote the G. And then we have guanine, guanine is paired with cytosine. That's why we wrote the C. And then we have adenine, which is paired with thymine. But remember, once again, we're using uracil. That's why the U. And then we have this, the third codon on the message is AAA, which is adenine, and we're just gonna replace it with U because we're talking about adenine paired with uracil, right? Then after we already have like the first, after the messenger RNA has the code, it's going to pass to the tRNA. This it's basically to ensure that the appropriate amino acid is inserted in the protein. So we're gonna be using the new code, the one that we already translate. So we have adenine, cytosine, and uracil on the first column, ACU. And we're gonna be and so we're gonna be using like and A goes with adenine goes with uracil, sorry. So we're gonna put like U. And then cytosine goes with guanine, so that's why we wrote the G. And then uracil goes with adenine, that's why we wrote the A. So that's how we do it. And once you have the whole code, we're gonna use the codon chart. The codon chart is this table that we had on the portfolio that I think you already know because well we're just explaining this to you teacher, so you of course you know what the codon chart is. We're gonna be using the codon chart to know what amino acid are we going to use. Like okay, we already have the code, but what with this? We're gonna use first. We're gonna use the uracil and then G and then A. So that's how it goes. So remember that the codon chart has this, it just goes like from big to small. So you just have to follow the codon chart and the name of the amino acid is going to appear. So we are basically going to be just like decodifying the message with the codon chart. And once you get the amino acid, we're gonna have this whole change. You have to do that with each codon, okay? So each codon is for one amino acid. They're all different. So that's why we get the whole chain of amino acids. And at the end, that's what the protein, that's basically like the synthesis of the protein. We're getting the protein. So the final product, it's gonna be the protein, basically. So there are some codons that are like, well, messages that are longer than others. If we go down, we're gonna see that we have, well, this one is this one is a short, the long one, sorry. We're just gonna do it once again, so to understand how this is done. Again, the DNA format message, we need it in the RNA message, so we're just gonna be replacing and doing the same exact thing that we just did. So first for mRNA, we're gonna take the first column, which is TAC, and We have to remember that thymine is paired with adenine. That's why we're going to change that. It's not going to be T anymore. It's going to be A. And then we have adenine. Remember that adenine is paired with uracil. So we're going to put the U in there. Then cytosine paired with guanine. That's why we wrote the G. And then on the second carbon, we have CTC, cytosine. So that's why we wrote the G. And then thymine, it goes with adenine. That's why we wrote the A. And so on. Once you have the mRNA, we're going to change it to tRNA. Remember to ensure that this is the proper amino acid. So we're gonna do it once again with the new code. We have the first codon, it's gonna be AUG. So we're gonna A, it's gonna be paired with uracil, so that's why U. Uracil is paired with adenine, so that's why A. And then we have guanine, which is paired with cytosine, that's why C, and so on. This takes time. And at the end, once you have the whole change translated, now we're gonna use a codon chart once again, and we are going to get the amino acid. Remember that I told you it's double A because it's amino acid. So we get the change of amino acids, we decode the message, final message, and then we're gonna have the protein once again. I don't know if you want to show like the peak of the codon chart at the end, just for the teacher to understand how we did it. Like we can continue with the presentation. Probably if we can at the end, we will show you how the codon chart works with. So now we go with Emilio's part.
Okay, so in the part of analysis and conclusion, we are presented to several cases or several questions, and we need to answer them. In the first one, that is apply concepts, it is how do you know which database must use when you transcribe to the DNA sequence to mRNA codons? If we consider that the nucleotide bases are adenine, thymine, guanine, cytosine, and uracil, they are found in RNA. Therefore, the adenine is paired with the thymine or uracil, and guanine is always paired up with cytosine. And inside the transcription, we can see that a single strand of DNA is transcribed in order to become RNA along with a respective base pairing, except that RNA doesn't contain thymine and it also uses uracil. And in the second case, that is suppose, we need to predict the DNA sequence for the first message in part B began with TGT, CGA, CGA, TGA instead. Is the message changing? Why or why not? So if we can go a little bit lower, during this specific case, during this specific case, the message will be forced to change because each base has a specific pair bonded to it. Due to its original standard matching sequence, it will be ACT, GCT. And if the case of the last letter of the first set of three numbers was to ever change, then the matching sequence will be ACA, GCT. And in the third question, we are found with use analogies. And we're presented with a case. So this case is we need to suppose some codons to different amino acids are mapped. What would be the effect on your translation of coded messages? What would be the effect on protein production? So when some codons are mapped to two different amino acids, the translation of coded messages would be wrong and inaccurate. Due to the mapping, some copies of different proteins will be created and most of them will fail. So in the fourth case, that is sequencing, we can see that during the real production of proteins in a cell, what could happen to an RNA chain before leaving the molecules? Before leaving the nucleus as mRNA, eukaryotic transcripts are modified or processed in different ways. Both exons and introns are transcribed from DNA to RNA. However, the RNA leaves the nucleus The nitrons are removed and the exons are joined in order to produce an mRNA molecule as well as continuous coding sentences. And in the fifth one, we need to evaluate and revise. What step could add after transcribing the DNA to develop a more complete model of protein synthesis? It will be transition, sorry, because it is a process of protein synthesis from mRNA protein synthesis that takes place on ribosomes. Therefore, the translation is a step that it is used to make a complete model of protein synthesis after DNA is transcribed. And in the sixth case, we need to create our own message. This part is going to be explained by my partner.
Okay, well, we already saw how this process of decoding messages was with Danna, but basically what we did here that we create our own message was first we pick different codons which are built as nucleotides of the DNA, and it's the chain of the DNA, and then we form the codons and created the first line, and then we had to translate it to the RNA messenger. Just as it's the exact same process. The only thing that we basically create was the first code of DNA, and then we just follow the process. And well, we got it to the messenger RNA, and we decode it, and then we inverse it to the transfer RNA, and we finally get the amino acids code with the codons chart that Danna told us about, and we're going to see a little image in a few seconds, sorry. And finally, in the amino acids, we got them from there, and we could get the proteins, and we finally decode the entire message, just as simple as that. It's a simple process, but it's kind of it takes your time. You have to sit down and do it, and take your time to do it. And now we're going to see the chart that I told you about, the codon chart.
Okay, so this is a current chart that we were talking about. After we have got the mRNA code, then we have the tRNA code. And once we have that one, we now are going to understand how this works. So let's just create a new one. Imagine that you have U, A, and G. In this case, that's how the codons are. You just have to follow the big ones, or no, it's just follow the codon. Just go with. We're gonna have, let's say, for example, UAU. This is one codon, and for example, we first focus on the U, the big one, and then you can see that there are like medium-sized letters next to the big one. So in this example of UAU, first we focus on U, and then we're going to move with A. And then the second one that follows is U. And there you have it, it's tyrosine. And that's basically how you do it. You just follow the codons in order. Imagine like the first letter, it's for the big one that appears in the center. The second letter, it's the one that it's next to it, and then the tiny letters which is the last one in the sequence of the codon. It's the one that is going to tell you which amino acid you're working with. So basically, this is what happens. After having our tRNA message, we just have to use a codon chart to know which amino acid we're using, and then you have your change of amino acids, and there you have the protein. So we just wanted to show how we did it, but that's basically how it works.
And we also have another chart we don't have right now, but that chart is when you have the amino acids, it gives you the letter that corresponds to the protein, so you can finally decode all the entire message, but that's another one. But yeah, as my partner says, this is basically all what we did in a comprehension activity. I hope that's enough, and you can understand based on what we explained. And thank you so much for your attention.